First, it is my pleasure to introduce Christian Conrad, who accepted the offer to contribute to our program with his W2 professorship for intelligent imaging. Christian studied biology in Freiburg and then worked for 4 years with a startup company developing automated microscopy systems in cytogenetics and pathology. So he has a truly agile career development, moving from academia to business and again to academia, A to B to A. So the moving back to academia means that he's continued with his doctorate at the German Cancer Research Center in Heidelberg under Professor Roland Iles, where he studied automated microscopy and pattern recognition. He then went on to head various research groups on automated microscopy in his field of expertise, intelligent imaging, first at the EMBL, the European Molecular Biology Laboratory in Heidelberg, and later at the Cluster of Excellence BioQuant for quantitative analysis of molecular and cellular biosystems at Heidelberg University. In 2018, he and Professor Roland Iles joined the Digital Health, or formed the Digital Health Center of the BIH along with other colleagues, and he is now with us in this professorship since this year. Now, Christian Conrad introduced machine learning into his field of medical image analysis early on. So he's not just a follower, but can be considered an influencer also in the use of artificial intelligence in his field. So in effect, thousands of cells and samples can be processed for pattern recognition using high throughput methods. With the combination of omics approaches, you get an even deeper insight into cellular phenotypes and functions than can be developed based on conventional microscopy. And thus he, among others, makes a very essential contribution also to large initiatives such as the Human Cell Atlas. So Christian, now it's your floor. We are looking forward to your talk.
Yeah, thank you for your introduction and the opportunity to present our results and workflows. And to the audience here, probably also the first question arises, what is intelligent imaging? And secondly, how is this linked to the Digital Health Center, BIH, or machine learning at Charité? And conceptually, I would like here to introduce intelligent imaging, which is an interaction of machine learning algorithms to automatically change and parameters configuration of screening microscopes while scanning based on a classified event. In this example, for instance, this is mitotic metaphases to generate high-resolution time-lapse movies to save time and storage capacity. And consequently, we have further applied this feedback microscopy, mostly confocal, to FRAP and FCS, two advanced fluorescence microscope methods for quantification of diffusion and concentration measurements. Now, ideally, mitosis look like this, labeled with DNA, chromatin, and the cytoskeleton marker, tubulin and actin, beautifully exhibiting prophase when microtubule growth from centrosomes take place. In metaphase, chromosomes are aligned and attached microtubules drag sister chromatids apart during cytokinesis. Cell abscission will be completed. However, in genome-wide mitosis RNA screening conducted at EMBL in Heidelberg, we faced many more complex chromatin phenotypes. And at this time, we used support vector machines as trained classifiers from extracted nuclei features to determine the separating borders between phenotype classes. Now, all this roots back to preliminary work in automatic classification of subcellular phenotypes, benchmarking different machine learning algorithms at the German Cancer Research Center, and when artificial neural networks were not deep at all, but very shallow, handling only 24 extracted features in one hidden layer with 10 nodes. And support vector machines at this time still outperformed any neural network. Today's deep learning includes intrinsically feature extraction with thousands of inputs in multi-layered convolutional neural networks. And this would have been impossible without the development of graphical processing cards, training models within hours instead of days or weeks in the past. But not only the computer algorithms and the hardware performance dramatically changed, also the biological side altered a lot. So 3D cell culture organoids, as we have seen, became accessible as routine in vitro models. These are MCF10 chromatin labeled spheroids captured for a solid phase reverse transfection screen. And these screening acquisitions in time-lapse are not possible with confocal point scanning microscope anymore, because phototoxicity and the acquisition time would be magnitudes too high. Our dual view microscope geometry, swapping between illumination and detection with two objectives, leads to almost isotropic reconstruction, shortly animated here. But on the other hand, generates terabytes of data. In our current SPIM system at BIH, we can even slide the specimen in a stage scanning mode to further maximize the acquisition rate. And Björn Eismann successfully conducted such a perturbation siRNA screen still at the German Cancer Research Center in Heidelberg. As a side note, throughout the presentation, I would also like to assign alumni to their current workplace to document train and also the opportunity in our group. So Björn automatically captured more than 200 spheroids in 24 hours time lapse, amounting here to 74 terabytes. But not only applied the presented deep learning strategies include feature extraction, here showing mitotic phase classification, again in time, prophase, metaphase, ana and telophase with the labeled colors. And yet also outperforming clearly, for example, the presented support vector machines shown in these confusion matrices and the 12% better accuracy overall. So moreover, deep learning was also used to segment masking objects, again, nuclei, using an algorithm which was originally developed by social media to track sport events. So these trajectories conceptually outlays the different possibilities and correlation of modalities we can envision. So mini-bulk, single-cell, full-length RNA sequencing so far. I introduced just the upper branch here. It combines the imaging, multiplex, and the single-cell sequencing with recent development in the spatial domain, which would allow also intermediate measurements. All this might be obvious application in preclinical and precision oncology or personalized regeneration. And in a parcours de force of projects, I will very densely highlight the portfolio of techniques now. So here, unraveling the mitotic protein network by multiplex antibody staining, targeting systematically the same regulators using drug inhibitors. And probably this is the most complex experiment we ever did in our lab, performed by Lawrence Meyer. Stefan Thirier used combination of mini-bulk and single-cell spheroids linked also to visual morphology to identify an important lowly expressed transcription factor PROX1 here in more aggressive forms of colorectal cancer, which we actually missed in a single-cell RNA-seq experiment. Julia used biobank ovarian cancer organoids 3D, outperforming 2D cell cultures to retrospectively test 2D different compounds at different concentrations. And for wider dissemination, we applied a specific simple fluorescence assay to study cytotoxic and cytostatic effects of these patient lines and could also give therapeutic advice for patients. With identical visual testing, but combining with single-cell sequencing of 18 patient-derived pancreas adenocarcinoma PDAC organoids, here Teresa observed seamlessly transition of heterogeneity of previously reported PDAC subtypes, namely classical and basal-like subtypes. And all these efforts in single-cell technology, we entered the pancreas field from the Human Cell Atlas Initiative, making the use of an 165-year-old single nuclei protocol, thanks to Luca Tossi, and added spatial cell maps with novel in situ sequencing, which is in principle sequencing targeted directly in the tissue sample. And in 2020, during the pandemic, our lab was almost completely drawn into COVID-19 research using single cell and nuclei sequencing. And we could, as early as May 2020, release first insights of cell types expressing ACE2 and TMPRSS2 host factors, particularly in transient secretory cells, here shown also in the upper part. Lawrence and colleagues analyzed similarly severe COVID-19 patient and explored the epithelial immune interactions of the respiratory system and identified, as depicted here, CCR1 as one central cytokine promoting adverse signaling in macrophages. And you might have seen or heard about the following clinical study for this target in an extreme short period. And I think the pandemic made these collaborative processes very agile and pragmatic in 2020, allowing this quick interaction, the analysis, the interpretation, digital and online, which was most likely essential for the success of this study. And personally, in the future, I would favor such online incubator space for other translational tasks. I would just state here. So here, as a short summary, the classification, the organoid work, the single cell sequencing, also with key publications. I added also recent COVID post-mortem publications, and as you see here also in Nature and Cell for completeness. And I would like to highlight the collaboration with the neuropathologist Helena Hartburg and Frank Heppner in this context. As outlook, I would like to close the circle here with an arrow which looping back to the classification task and deep learning, and urgently needed also in spatial transcriptomics, or we could say back to the roots. In the context, I would also motivate the embedding of biological pathways and networks. Here, Zoran Lukassen, now junior group leader in our center, who modeled the potential biological connections in a deep learning autoencoder to discern the genes, the co-expression, and the pathways. So my thanks goes to everybody in the lab and so many collaborating partners and co-authors, where we constantly and also intentionally swap between the wet and the dry lab. And I thank you all for your attention. Thank you very much.
Yeah, great. Thank you, Christian, for this very concise but still very rich talk. We have enough time for questions. So if you have any questions from the audience, please raise your digital hand or leave us a note. So maybe I can start with your remark. You would be interested in promoting this format of the online incubators for translation. That is how you call them. So this, what exactly do you have in mind when you think of this approach?
Well, I think there are collaborating platforms which we used. I can name some of them, but I think there are many by now. This was actually not only because of distance, but also of exchange, immediate contact, very important. And I think we talked about this before. I think such an I call it now incubator space. I could call it collaborating software, which I think was used in this pandemic situation, but could be used also for any other collaboration and communication within the BIH.
Yeah, absolutely. This is a very important suggestion, and we will have to identify suitable formats. It could also be used beyond the BIH, of course, for collaboration with other investigators outside, including industry collaborators. And that was another very interesting aspect, not only of your career, but also of the careers that you indicated for team members. So is it on purpose or more or less a direct consequence of the work that you are doing, that co-investigators, team members have a direct interaction with industry and eventually also move to industry from academia? Is that something that you develop per design?
I think this is intrinsically by my career, yes, one point, but also I think that it's that, of course, these people are attracted by, or the industries are actually contacted them mostly in these collaborations, and we get these people actually drained from our lab. So I'm not always happy about this, but of course that's also that's the consequence of being so closely collaborating with industrial partners, I would say.
And for some aspects of your work, you have real technical challenges. You mentioned the graphical processing cards. So I assume that in this case, you need industry collaboration to have the best possible equipment available for your research.
Very true. And I think this is also, and I mean, we are very well equipped with NVIDIA, for instance, graphical or GPU clusters, which is really a surplus here also at the BIH. But for the future, I think we have even more request and demand. And I think this would be an important asset also in the future to have this, especially
another tech. Sorry.
Yeah, especially if the data becomes bigger and even bigger, and even if we use feedback imaging or feedback microscopy.
And that would lead to my final question that the final question that I have in mind. Now, with the challenge of these large data volumes, are there approaches such as the MP3 approach to reduce data to the essential, so that you can reduce data amount by several orders of magnitude for digestion?
Yeah, that's, I think, exactly the idea. Yeah. So you would not only save storage, you would concentrate on the most important entities or data points, and this is, I think, the whole idea behind the imaging. I think it is most obvious in imaging, but it's obviously also needed in many other technologies and workflows. Yeah. So it's actually,
yeah,
it's actually also very known in physics, for instance, where also during data generation there's already a classification and selection of data.
Well, thank you again for the talk and the discussion.